In a laboratory in 2019, scientists placed living retinal neurons under blue light for 2 hours. Afterward, one key marker of DNA damage in those cells had jumped more than 4 times higher than normal. The neurons were dying. Somewhere between that finding and the headlines you may have scrolled past, a much bigger claim was born, that the phone in your hand is quietly rewriting the genetic code inside your eyes. It sounds like exactly the kind of thing worth worrying about. So we checked.
Here is the real question hiding inside that scary headline. Does the light coming out of an ordinary screen, held at arm's length for a few hours a day, ever come close to the light that broke DNA in that laboratory dish? To answer it, we need to go inside one of the most demanding tissues in the human body and understand precisely what blue light can and cannot do to it. The retina is not wired like the rest of your nervous system. Almost every other nerve fiber leaving your brain gets wrapped in a fatty insulating layer called myelin, which speeds up its electrical signal. But the nerve fibers of the retina, called retinal ganglion cells, stay bare for the first stretch of their journey, because that insulation would scatter incoming light before your eye ever formed a clear image. In exchange for a sharp picture, these neurons carry their signal the harder, slower way, which burns enormous amounts of energy. To keep up, they are packed with more of the cell's fuel-generating structures, called mitochondria, than almost any other neuron in your body. That is the first clue to why this tissue is unusually exposed. It sits directly behind the lens, exactly where incoming light is focused, and it is one of the hungriest tissues you own.
This is where blue light enters the story. Visible light spans a range of colors, and the shortest, highest energy wavelengths sit at the blue and violet end, roughly 400 to 500 nanometers. The lens and cornea at the front of your eye are excellent at blocking ultraviolet light, the even higher energy radiation behind sunburn and cataracts. Blue light, though, slips past that filter almost entirely and travels straight to the back of the eye, where it meets an enzyme inside the mitochondria called cytochrome C oxidase that happens to absorb light most strongly in that same range, somewhere around 440 to 450 nanometers. When a photon of the right energy strikes it, the cell's fuel-making machinery briefly stalls, and that stall floods the cell with unstable, reactive oxygen molecules. This is where the study picks up the thread. Once a retinal neuron fills with these reactive molecules, its mitochondria begin to fragment, splitting apart instead of staying fused into long, healthy networks. A fragmented network cannot keep the cell's cleanup systems running, and the extra reactive oxygen eventually reaches the nucleus, where it can crack open both strands of the DNA double helix at once. That is the 4-fold spike in damage the researchers measured, and it is genuine. Neurons in that experiment were far more vulnerable than the retina's support cells, and the ones tested inside a living eye, not just a dish, showed the same broken DNA and gradual retinal thinning over time. So the mechanism is real. Here is where the viral claim runs into trouble. And if you take one number away from this video, make it this one. The blue light in that landmark study measured 900 lux, delivered continuously directly onto exposed cells for 2 straight hours, with no blinking, no looking away, and no cornea or lens softening it first. 900 lux is close to the brightness of a heavily overcast sky. A phone screen at reading distance in a normal room delivers a small fraction of that, and a human eye blinks around 15 times a minute, constantly interrupting any exposure.
Direct sunlight, for comparison, can reach 30,000 to over 100,000 lux. If blue light intensity were a staircase, that laboratory damage sits near the top. Sunlight sits even higher, and an ordinary phone or laptop screen sits many steps below both. This is not a fringe opinion. The American Academy of Ophthalmology states plainly that there is no scientific evidence that light from computer or phone screens damages the eye, and it does not recommend special blue light blocking glasses for that reason. The tired, gritty feeling people describe after a long day of scrolling is almost certainly ordinary digital eye strain, caused by blinking less and staring at a fixed distance, not a photochemical injury rewriting your retina's genetic code. If you were bracing for worse news than that, stay with us. The real risks to your eyesight are still coming, and they are more interesting than the myth. Dismissing blue light entirely, though, would create its own myth, because for some people, the calculation genuinely changes. After cataract surgery, the eye's natural lens, which normally absorbs a meaningful share of high-energy blue and violet light, is removed and replaced with an artificial one. Depending on the replacement lens, some patients gain more than double the blue light exposure a 10-year-old's natural lens would allow. And in elderly patients whose natural lens had yellowed with age, exposure right after surgery can jump by as much as 7 times. Welders and anyone who has stared too long at the sun can suffer a genuine, documented photochemical burn on the retina, sometimes from a single exposure. These are the situations the original studies on retinal light damage, dating back to the 1960s, were actually built around, decades before anyone owned a smartphone. Even among scientists who study lifetime sunlight exposure and long-term retinal health, the picture is not fully settled. One long-running survey of Chesapeake Bay watermen found that those with advanced macular degeneration had received significantly more blue light exposure over the prior 2 decades. A separate major European study found only a weak association that did not reach statistical significance. A group of ophthalmology researchers argued in a recent journal article that the blue light hazard concept has been misused as a marketing strategy to sell people glasses and lens implants they do not need. When specialists who have spent careers on this exact question still disagree with each other, that is usually a sign the honest answer is uncertainty, not the internet's confident version. So what actually moves the needle on your retina's long-term health? Genetics first. A gene called complement factor H, identified in genetic studies published in the mid-2000s, is one of the strongest known risk factors for macular degeneration, and no amount of screen time changes what you inherited. Smoking is the single strongest modifiable risk factor, raising the odds of vision-robbing degeneration far more than screen exposure has ever been shown to. Diet counts too. A major nutritional trial funded by the United States National Eye Institute found that a supplement formula built around antioxidants found in leafy greens, including lutein and zeaxanthin, lowers the risk of progressing to advanced macular degeneration by roughly 1/4 in people who already have the intermediate stage of disease. And new drugs designed to directly protect the mitochondria inside struggling retinal cells are now in late-stage human trials, though the most closely watched one narrowly missed its main goals in an earlier trial and remains unproven. None of this makes blue light boring. A tissue thin enough to be almost transparent, wired directly into your brain, running some of the hungriest cellular engines in the human body, sitting exposed behind a lens that focuses light onto it every waking hour. That is genuinely one of the more delicate arrangements biology ever built, and it deserves real protection. Sunglasses that block ultraviolet light, regular eye exams as you age, and staying away from cigarettes.
What it does not need is a myth borrowed from a laboratory dish and pinned onto the device sitting in your hand right now. The next time someone tells you your phone is rewriting your DNA, you will know the one question that actually matters. At what intensity?